Okay, very good afternoon. So, a very warm welcome to all of you in this seminar. And before we start seminar, let me mention or introduce to our two very eminent guests and collaborators and our very good friends. So, one of them is Dr. Steve Brown. He is the director of the Peanut Foundation based in Georgia in United States. And other very good friend and collaborator is Dr. Baozhu. He is research scientist at USDA-ARS. Anyway, he will be talking or I will be giving an introduction in a minute. So please join me in welcoming to both of them to ICRISAT. Now, let me mention, and you already aware, many of you may be aware that few days back on 22nd of February, we had this announcement of the peanut genome sequence paper in Nature Genetics. So this work was mainly coordinated by Peanut Foundation, and I think Baozhu is going to present something, this thing that International Peanut Genome Initiative. So this is coming basically from the coordinated efforts of the Peanut Foundation coordinated by Steve Brown nowadays. So congratulations Steve for that and to the entire peanut community basically. And we feel very privileged to be the part of that consortium. Now coming back to giving brief introduction about Dr. Baozhu. I think this name is not an unfamiliar name to our group here at ICRISAT because of several reasons. One is that he's long-term collaborator for us. And second is that some of our scientists, they have stayed 6 months to 1 year time in his lab. Third is that right now in our group, even here at ICRISAT, so in the peanut genomics people, we are having 6-7 people. They are doing more work for Baozhu than they are working for us. So, so everyone knows about Baozhu. And at personal front, I would like to say I enjoy a very good relationship, very nice friendship with Baozhu. He is a great friend. Whenever I go to US or China or anywhere, wherever I meet, I always feel very excited and very nice interaction with you, Baozhu. I mentioned that he is a research plant pathologist at USDA-ARS and at Tifton campus. And his research interest, basically, he is doing a lot of work in molecular genetic studies of corn, earworm resistance, and development of markers for using marker-assisted selection. Also, mechanism of resistance and genetic studies of resistance to aflatoxin contamination in corn and peanuts. So his laboratory is working on both aspects, on both crops, corn and peanuts. He did his PhD from Louisiana State University and before that masters from University of Kentucky, and then he did his BSc in Shanxi Agricultural University in China. His laboratory is developing and applying advanced biotechnology tools to identify genes for resistance to insects and aspergillus flavus in both in corn and peanut. And also, as I mentioned earlier, he's developing markers associated with resistance to assist in transfer and selection through molecular mapping, molecular genetic studies, and characterization of biochemical and molecular basis for QTLs involved in resistance. And he has done wonderful work during last few years, and I know Baozhu from 2007. I joined ICRISAT in 2005, but then I attended the meeting in Atlanta. That was maybe second meeting of the, yeah. So from then we know we are good friends. And after that we have been involved or collaborated on many projects. So he's visiting us with Steve. So we thought it will be good. I'd get to have the updates. And I have also requested Steve to give a presentation on Monday, which he has kindly agreed. So today we will have the presentation from Baozhu, and on Monday we will have from Steve. So with these words, please welcome both of them once again and welcome Baozhu for the presentation. Please come.
Good afternoon. It's really nice to be here. Just to chat, to chat with friends about the science. We started yesterday with the DG. We had a really good discussion about the science, about the international collaboration. And also yesterday afternoon, last night, we had also good discussion about politics. Not just science, really not something about politics. Interesting to know that Those two represent you guys here, young generation. And Steve and I really kind of old, old generation. One thing I guarantee you, last night we discussed, those two, they believe in China. They believe in China. I believe in, listen, I believe in India. Okay? The reason why democratic society and a communist society at politics, but just remember that down the road 20 years, 10 years, 20 years, 30 years, you're going to pass China. Let's move on. That's politics. I used these slides last year. You know, talk about orphan legume genome whose time has come. You know, a story about a peanut genome. This really is an international effort. And that's the title today. I want to really demonstrate the international effort to tackle peanut genetics and the genomics. I want you young generation to learn the international effort is so important. So not just do your own thing. Get out your office, your lab to find a partner, to find collaborate. Really can make good progress. Okay? I always use these slides. You can see here, Georgia, USDA, and the USDA. In my talk, 99%, I use this. Okay? And this is something I talked about last December. This collaboration among Georgia, USDA, and the USDA, we have mission. Our mission is to find a candidate gene, to develop friendly marker for breeders. The purpose is for marker assisted selection to help them, the breeders, to breed, to improve the peanut and other crops, you know, quicker and better. Also, don't forget, we as scientists, particularly international collaboration, we're also looking for the future. That's for the future, magic in Mars. You know, don't just do your today, your work, also looking for future. Okay, so I like to use this one as example. You know, that's what I gave last December, a progress report to Steve Brown, the Peanut Foundation. Before everything, I want to talk about these three parts today. I like to introduce myself. You know, just myself, my research, my background, my interest. I like to just use this International Peanut Genome Consortium, this sequencing Rajiv just mentioned, the paper published finally. But it's a long way, it's an international effort. I'm going to give you some historical events, such as 2004, 7, 10, 12, 14, 15, and 16, and beyond. Then I can give you exactly the collaboration between USDA and the USDA. That's me. Rajiv just mentioned, I got my master's degree in 1983. Just remember 1983, that's a third classes after 10 years Chinese cultural evolution. Those 10 years of no university education. So after that 10 years, 1983, 81, 82, 83, that's 3 classes. So I got my BS in 1983. Then worked in China about 6 years. I'm working on millet. That's a millet. See this? Foxtail millet. Then went to US, 1992. I received my master's degree, then PhD in 1995. Worked 1 year in New Orleans as a postdoc, then moved to Tifton since 1996. This is my family. This is my younger daughter, Victoria, my wife, Jinping, my son-in-law, Jake, my oldest daughter, Susan. There's a Buster, my dog. There's the 2 dogs from my daughter. There's a cat, also my cat called JJ. Percy and a Huckleberry. There's my family. That's a new addition. That's Maggie Sue. She's the best. That's my family. My research, Rajiv gave you a brief introduction. I'm working on corn, peanuts. Those 2 crops, aflatoxin is linked. That's why I'm working on those 2 crops. In addition, also I'm working on other diseases of peanuts. Aflatoxin, we all know that, so causes liver cancer. And this is in these regions, perennial risk from north 35 and south 35. This India, Hyderabad, somewhere here, so in the middle of this aflatoxin zone. But I wanted to point out is that in 1977, for example, US aflatoxin reached the Midwest corn belt, they call it corn belt, because of drought stress that year. Since then, we study aflatoxin, drought year, they really can expand this one. And those hot spots, Africa, South America, Central America, Southeast Asia, those hot spots are linked to a lot of diseases. This slide, is just showing that liver cancer and high risk of aflatoxin. So they just correlate, very high correlation. To deal with this one, United States had this USDA aflatoxin elimination annual workshop since 1988. Every year until 2007, somehow that's the last year. I went to this meeting, attended this meeting since 1993. Every year I was there. And also the 2007 was the AAGB, second AAGB. That's where I met Rajiv in Atlanta. I want you here to remember when aflatoxin is 20 parts per billion. That's where the limit. Over 20 parts per billion, you cannot transport. Okay, how much is 20 parts per billion? I want you remember 1 part per billion equivalent to 1 second in 32 years. Really, really very, very small amount of aflatoxin. 1 second in 32 years, you have 20 seconds. You got it. Next, just wanted to show you this international peanut genome consortium. Okay? Why are we doing that? We all here are scientists, agricultural scientists. Our sole mission is to produce enough crops to feed the people, right? That's the bottom line. The goal is 2050, there are going to be 9 billion, 10 billion people. That's a huge challenge to us. Another challenge is currently, today, the hunger and the malnutrition. You kill the child, 1 child every every 5 seconds. One child died, so another 5 seconds. So we really have a big task. What we can do for peanut? So those peanut production really associated with those areas. So we can grow more and better peanuts to feed those people. Once one peanut, because protein, high protein, 26%, oil, 50%, 50% oil, 25% protein. So those can help those people. Challenging to us, 73% area, those 70% area only produce 45% peanut. That means yield. We have big room to improve the yield for those areas. Aflatoxin. You may recognize this guy from just from the back. Valia, Fred. When he gives these slides, Bamako, you can see this groundnut seed and the groundnut paste. All those they purchased from the market. Every single samples. That's a 20 parts per billion. Every single samples over 100 parts per billion. That's the challenge to us. How to help those people to reduce the risk. Second challenge for us as a scientist is the complexity of the peanut genome. Once the size, the human genome is about 3G. So peanut genome about 2.8G. So just like a human. That's a challenge, size. Second challenge, polyploid. So peanut has A and a B genome. So that's another challenge. So that's why we had this International Peanut Genome Initiative. Under there, there's a Peanut Genome Consortium. Once executive committee and at the administrative office, that's in Dr. Steve Brown's office, manage this project. That's so many participants, different institutions, and different people, also from China and the USDA. So they are 135 members from 79 institutions in 20 countries. Okay, so really wide, worldwide effort. This is the committee. Dr. Varshini and myself are in the committee. This committee led by 3 chairmen: Scott Jackson, Peggy Alzea-Akins, Richard Mitchell. This contract signed on March 28, 2012. This contract was signed on March 28, 2012. So that's a summary. That's a long way. Really, for peanut genome, started 2003, a group of people just talk about legume. Let's have something for legume. 2004 really is the first time in Atlanta talk about U.S. Peanut Genome Initiative. First time, 2004 in Atlanta. Then 2006, we had the first AAGB meeting in Guangzhou, China. The second AAGB meeting in Atlanta. Then continue, third one, USDA, Bamako, Mali, and Brazil. Then we're back to China, then back to US, Savannah. That's AAGB meeting. During those years, one thing I want to point out here is the first peanut genome project 2010 inaugural meetings December 2010 Atlanta. Before this meeting, Victor, Howard, and I went to China to talk to the colleagues in China if we want to start this project. That's September 2010. From there, 2012, we launched the peanut genome. We signed the contract with BGI, and launched this sequencing project. Then 2014, April 2nd, 2014, the sequencing data released to public. What else? In 2015, November, we had the 8th AAGB meeting, 2015. In 2017, March, next year, March, going to have another AAGB meeting, 2017, in Argentina. What do we have this? February 22nd, Rajiv just mentioned. The paper published on Nature Genetics. So that's this paper. This paper was received July 30, 2015, and accepted January 29, 2016, and published February 22nd, 2016, this month. So we really celebrate. We took this shot as 2014 November in Savannah. Celebrate. The people, they are crazy, but I think it's worth to celebrate. Okay. So as you can see, this international effort really long way, comes a long way. So it's not simple. What are we going to do? What are we going to do beyond this base pair, A, B, C, D, A, B, C, D? What are we going to do? So this collaboration, USDA and the ECRSAT, the collaboration to take this sequencing to the field. That's what we do. How to do it? First, still asking, always people talk about S and T. That's S population and T population. From Sound Lake 97R and NC 94022, and the tip run across to GT-C20. That's a long way too. 2005 and 2013. During those years, many papers published, and also those two populations, S population, there were 143 real lines, T population 248 real lines. In there, 143, 118, those lines are sequenced, resequenced. So this Sound Lake 97R and the NC 94022, you can see phenotypically very different. This one's tip runner GTC20, also very different. So we use this particular T population, we study aflatoxin. That's in the field, that's in the laboratory. You can see tip runner. After 7 days and 14 days, you can see the fungus, how the fungus to grow on these kernels. This is GTC20, 7, 14, and 21 days. You can see this one does have some resistance in the laboratory. So this is the first joint international effort led by ECRSAT, Rajiv, developing this international reference consensus genetic map. And the Tifton, those S and T are part of this. This is the second international high density reference map. And the S and the T maps also are part of this. So this international effort. For all those works, hard work, you spend the time in the field, phenotype. This is my colleague, Dr. Albert Colbert, spent hours and hours from 2009 to 2013, 2014, doing every year, doing spend hours and hours doing the disease score. See, it's an early leaf spot, and you can see the early leaf spot and late leaf spot. Sometimes they mix, sometimes you can separate, but Dr. Cobras is a plant pathologist. He knows clearly help me to separate those two diseases. Sometimes it's very hard. This one, for example, 2013 early leaf spot. You can see almost 100% early leaf spot. From the back and the front and the close up. And when you spend the time in the field, you can see something else. That's why I encourage you, scientists, to go to the field, not just in the laboratory. In the field, also we see this leaf copper segregating in this population. Otherwise, we don't have this phenotype. So we also collected some phenotype against this trait, leaf copper. This Dr. Cobrath and Dr. Ni helped me to score this trait. During those years from 2009 to 2014, we scored biotic stress and oil quality, morphological traits, and also yield components. Particularly, for those diseases are important, and also for the quality. So those already published. So we are working on other traits. This is just a example publication. You can see Penny's name first here, and also Owen here. So really, it's showing the collaboration. And just like I said, I repeat you this example. I want to talk about the detail about the candidate gene, the markers, and some magic in the mouse populations. Again, you see the S and the T, and talk about the future, magic in the Mars. That's the future. Also, I like to touch a little bit the NAM, the Tilly. This NAM, the Tilly US, really we had this before. If we do not look into this, kind of people just forget. So basically, we just take advantage of the resource we have to see what we get. And for the S and T, we had the first generation map, second generation map led by Manish Pandey. This one I call third generation map, led by Owen Carroll. And we're going to have the resequence of the S and the T. That's what's his name? Going to, basically going to lead this part. So we're going to have a fourth generation group. Yeah, by the resequence. Okay. For example, for the S population, this we submitted 2015 in revision right now. That's led by USDA. Poland, of course, Poland is not here anymore, but I still credit to USDA. I just want to give you to summarize what we got for disease resistance for the S population. Look at this. For TSW, that's the #1 disease in Georgia. We have QTLs up to 30%. It's really high. For early leaf spot, up to 30%. Late leaf spot, we have QTL 17%. So those also you can see, those disease QTL, they overlapped on A1, chromosome A1. And for the late leaf spot and some early leaf spot, primarily on chromosome B03. Really interesting. So, you can see there's something there we can call gene, but we need to identify that gene. I just give you one example for the S population. You can see the QTL, and that's 2 years, three different data sets, and folks, you can see QTL in this region. Also, we genotype those with, if you have this gene allele from Sound Lake, they are susceptible. If you have allele from NC94022, showing resistance. The reason why I'm showing this, because we're going to use this resource to develop Mars population. For the T population, we done the TSW in the leaf spot. Here I'm just showing you aflatoxin, because for aflatoxin in peanut, there was no paper, no QTL so far. I think we got some. One is just for aflatoxin contamination. We have one QT on chromosome 4. It contributes about 10%. And that's the one we identified resistant gene in that region. Another one for the fungal colonization infection. There's one on chromosome 5, contributed 14. In this region, we identified one single resistant gene. I just got this data last night. So here you are. For this, from A chromosome 4, has 18 resistant genes, a QTL, for that fungal colonization. One here, for this aflatoxin, last slide, you see, it's one single resistant genes. And for one marker, we can see 3 times. That's what I'm talking about. That's a QTLs. This QTLs analyzed by Manish Pandey and the EPRSAT group. That's what I got last night from my student. Just did a quick comparison. What do you see here? This QTLs, they identified one resistant gene between those two markers. So this marker had 3 hits, but this one single resistant gene. I want to show you another collaboration between USDA and the EPRSAT. Jake, he done three months of work last year, generate ton of data. I really appreciate the Passat helped analyze those data. So what we did last year, in general speaking, is just try to find out why Aspergillus flavus produce aflatoxin. What is the role of aflatoxin? If you produce aflatoxin, what is the role? And what's the biological function? So we had 3 aflatoxin producers, 3 non-producer, 3 H2O2 concentrations, 2 media, yes sugar, and yes peptide. And total 64 RNA samples. That's a lot of samples. Based on those PCA and MSA, I don't understand this, analytical term, but you can ask. That's down here. What I'm saying is that it's really interesting, separate by different media. So I call it carbon source specific. That's the plan. We talk about the data analysis and the results. Really, it's just 4 papers, 4 manuscripts going to be, prepared. One overview paper already complete. Target is Nature Communication. Jake going to working on this one. Spooth is working on this one. Manish working on that. So you can see it's also a joint effort, not just for research, also joint effort for the final publication. Add on this paper, we already lined up, to do the proteomics to confirm what we saw for the RNA-seq. That's the one we're going to have 24 samples. You just see this 2D protein. You see some difference. You can see there, you can see here. So we have 3 acids. That's the concentration of H2O2. And going to be ATRAC and the 2D. ATRAC already started last Friday in Florida, Gainesville, University of Florida. This 2D page going to be done in China. That's Jake going to China, working on this 2D. I want to move on, just say why we prepare for next, generation 19 populations. It's so important. The reason why the International Genome, Consortium is working on, develop the markers and sequencing, but they need the material to work on. So for me, and also said, we prepare for future. That's the Winston magical population. Magical population really is a multiple parents. So what we talk about the real is 2 parents. Here's the multiple parents. I want to use the example 8 different parents. You're making F1 cross, the 4-way cross, and 8-way cross. Finally, you put all the 8 parents together into one single F1. Then develop. Goal is 1,500 lines. Then to F3, hopefully we reach F3, F4. That's where we start in genotyping and the phenotyping. So what we put here in Tifton, that's 8 lines. The first 4 lines, we talk about those 2 reals, S and the T, because we have a lot of information. I include the 2, you heard about the caps, and also I'm going to talk about those 2. That's a NAM, and the plus 2 newly released varieties in Georgia, with the Georgia 13M and High Lake, with tip NB High Lake. This one has High Lake and also resistance to nematodes. That's the 8 line I selected. For the Mars, same principle like a magic, but those 8 lines is selected from S population. So we have those S population selected, those 8 different reals. We know those reals are High Lake. We know their oil content from low 43 to 51, from low to high. And also we know their QTLs. The QTLs to TSWA, the QTLs to early leaf spot and the late leaf spot. So hopefully through the Mars we put all those traits together. High Lake, high oil, low oil, resistance to TSWA, leaf spot, all those three together. So that's the status in Tifton, the magic. We done the 2-way, we done the 4-way. That's the pass we produced 4-way crops. So we are right now probably already planting these seeds, prepare for 8-way crosses. So we do check the F1 if they are real F1 or fake F1. That's in the greenhouse. That's F1 for prepare for 4-way crosses. So it's done. I want to talk about the NAM about a few minutes. NAM. We know the NAM is use common parent across different lines of founder. If you consider one by one, it is a real, single real. That's the population developed in US called CAPS. So NAM, if you do the one single real, you can identify QTL. The second real population, identify another QTL in similar region. You keep it going, hopefully we can identify really, really narrow, refined QTLs, accurate location. That's the NAM, multiple population. just give you some background about the U.S. CAPS population. It's accordingly published this paper in 2013. Just introduce, this recombinant inbred lines populations for peanut. What he did is developed 16 different real populations. This 16 from 2 common parents times 8 different parents, 2 by 8, so total 16. That's 2 common parents and 8 unique parents, 2 by 8. So they selected those parents based on their different resistance to different diseases, and also disease and the market type and how late. So really diverse lines. So what they did, they separated into 2 sets. One set A and set B. So 2 by 4 and 2 by 4. So what I did is I choose this one, 2 by 4, set A, to see how powerful the NAM. You can see here, that's the proposal. So they developed these 16 real populations. What they propose to do by individual labs, if they are interested in different traits, they just study the individual population. If you study the individual population, just like one real population. So what my job is, my interest is to work together to study the whole thing. So we call it NAM, working in collaboration with ECRSAT to see the whole all together as a NAM population. So just showing this in this NAM, the Florida 07 common parent and Tifton as another common parent. 4 different unique parents. You cross, you produce each line, real lines. It's when you look all 4 together, it's a NAM, Tifton NAM, and another one 4 together, it's Florida 07 NAM. So basically, I put all they used to call CAPS. I put all this together last year. I went to court. I just asked them, you see, if we can look all together, see what's the power can be compared with the individual real population. So we had 1133 lines plus parents, total 1149. And we planted it in June 15th. That's this field. Really nice, this drone. Use the drone, you can fly over, take this shot. Each plot is 2 rows, 5 feet long. And this alley between plots is 10 feet. So 10 feet alley, 2 5-foot row plots. Just give you a shot in the middle of the season. You see the diversity, segregation for the morphological traits, for disease. And then you see another shot, the size, see the size, tall and the short. This is a shot before the day before digging, before harvest, the day before harvest. You still can see some green, some totally defoliated. You see the disease segregation basically. Just see the trait we collected last year. Leaf width, leaf length, main stem height, plant size, leaf spots. Leaf spot we combine the early leaf spot and late leaf spot. And 100 seeds weight and 100 pot weight. You see all the average, you average it together. Just give you example, just for 100 pot weight. It's the difference from 129 to 170. Segregating them, just see those different populations. And you give you that range from 129 to 170, really big. That's good for us to study genetics. So did a quick t-test, all the traits. they are significant compared to those Tifton NAM and the Florida 07 NAM. Just see the segregation. For the leaf width, length, and leaf width, they segregate really good. It's nice. For the second NAM, also segregating pretty good. For the 100 pot weights in the Tifton and Florida 07, you can see they are segregating nicely. But I really wanted to show you the disease. We had a 2 reading, October 1 and October 26. You can see the disease progress really fast. In the first reading, you can see from 2 to 10. The second reading already progressed to 5 to 10. But if you compare those two populations, two NAMs, Tifton really the resistance is much, much better than Florida. You can see the number is 29% resistant lines, and here just 19%. Even before digging, they are still 6%, 5, and 6. Here only 0.9%, 5 and 6. Just what we're doing now, you just compare this and quickly do some heritability, broad sense heritability and narrow sense heritability. You can compare, just see the disease, for example. They're very similar. That means this disease is not influenced by the environment. But in contrast, you see the plant size, the narrow sense heritability, it's about half of the broad sense. That means significantly influenced by environment. Right. So if you see those NAM, Tifton NAM, and the Florida 07 NAM, of course you just see those numbers. You like to use Tifton NAM to select the resistance because the number is much higher than this. But if you see the plant size, of course, Florida NAM is much better than Tifton NAM. Every time I use the slide showing to credit those people, those people spend hours and hours in the field. Those 3 ladies, they collected all those trees this year. That's their credit. And I would just want to point out the SNP genotyping. Those genotyping going to be snipped genotyped by Semi-Arid that developed by E%. That's by E%. Give you a little bit of quick tilling. That's a tilling we planted in 2014, 2015. That's how you see it, segregated. The tall and the short, the main stem without a main stem, and the disease resistant, disease susceptible. Segregating that tilling. That's what why I took those seeds to the field. From Peggy, we saw this. 2014 and 2015, same thing. And you can just close up, you can see the size, tall, the short, big, and also small. That's how the tilling, they caused those mutations. So I shared this with Rajiv last year. Rajiv made this decision immediately. Again, that's a collaboration. He said, give me all those lines. I'm going to sequence the whole genome of these 34 lines. Those 34 lines. That's the DNA here. Secondly, it's a make F2 population. So I'm making 2 F2 populations. We're going to sequence it. That means we can identify that the gene clearly control the main stem, control the leaf spot. We saw those 2 traits. Maybe others too. Just in summary, S and the T population, we've done great. We've done great for the oil quality. We got 2 papers published, joined together. We got TSWV, early leaf spot, and late leaf spot. And also, we have 264 real genotyped by GBS resequencing. That's a group. To calling this. So you can see, the collaboration we can produce a ton of stuff. So we have this magic. We are in this stage, 8-way stage. And Mars also we're going to be in this 8-way stage. The NAM, you saw that's going to be genotyped by SNP, collaborating with UPRSAT. The Tilling also, the 34 lines DNA here. The F2 population is going to be ready later this year. During those years, we have a job education. Those kids. There's a Jake, PhD. There's a MS student. Those two, she's in army, in military. She's in medical school. Visitors. This last week we were in Florida, reporting. That's acknowledgement of many resource peanut foundation here, and also USDA. So without this funding, we cannot do anything. That's the people of 2014. That's a point here. At 2015, Rajiv stopped by Tifton. We had a lunch. Of course, Dr. Cobra. And thank you very much.
This is fantastic presentation, and Baozhu already has shown that how much technology and on top of that partnership or collaboration really contribute to the growth of the science and also development of the products, which is both scientific knowledge or publication at the same time also the products going to the field. And as he showed that together with Baozhu and RICRISAT and of course with other collaborators, a lot of things are coming. And Baozhu is on top of that. You already have seen the last slide that how much work we have now to do together. And yeah, good. So anyway, so thanks a lot, Baozhu, for great presentation and for all your collaboration and help. We can have few questions because there will be another presentation in few minutes. So we can have the seminar open for some questions. You have explained things so nicely.
No, that's just like in China. When you ask if nobody has.
Yes.
But in US, there's always.
Well, here also people will ask questions, don't worry. Yes, this is the one.
I have one general question. As we know that, and you mentioned that drought is the predisposing factor for aflatoxin. So how do you foresee the aflatoxin in future under this climate change scenario?
Actually, I have another slide just talking about the aflatoxin and the drought stress. I'm working on corn and the peanuts, also working on those fungus, Aspergillus. To answer your question, that's going to be a big issue because aflatoxin, this fungus, Aspergillus, is so unique, adapted to this drought, hot environment. It can survive, but other fungi cannot survive. That's why when you have a drought, the more drought you have, the more Aspergillus you're going to, So that's going to be a big issue. But so far, we do have the knowledge to deal with it, but the cost. For example, one thing I tell people, before harvest, you give water. Just one water, give it. When the crop needs water, give water. But we do not have enough. For example, just sit outside this building. All the grass kind of dying because we do not have water. Secondly, after harvest, you take our water away. That means the dry. You dry it and the storage, drying. So before harvest, give water. After harvest, take water away. But after harvest, the farmer
Sometimes they add the water on that kernel so that they can get the more price.
That's
probably
what
because,
in China I heard too.
Yeah.
They add water in there. When you add water, it totally destroyed. The grain only needs 3 days.
okay, good. Rakesh?
Very good presentation. So I'm curious to see the additive effects which you showed in some of your magic population.
Which effect, Rakesh?
Additive effect.
Additive effect
Values.
in that magic
Um,
population.
It is as high as 10 in one of your slides. So I'm just curious to know whether you plan to sequence that QTL and dig out some interesting candidate genes there.
Oh, of course, yeah. For the NAM particularly, talk about the NAM. The NAM are going to be genotyped by array, a slim array. That slim array basically theoretically covered all the genome, 58,000 slim array. So in that case, we really can identify the QTL or genes into that tree. It is additive, in fact, significantly.
And just to add that magic population is also planned for sequencing. So when you sequence, you will have the high resolution mapping. At that time, you can see what kind of genes. Okay, any other question? Okay, Rurkiwal. So unlike China, Rurkiwal is always having question. So even if you say normal thing that I'm going for lunch, he will say I'm having a question. So he will always got a question.
Thanks, Dr. Gove. Nice presentation. Based on explicit experience using Mars for chickpea, it was not much very successful. So what is your experience?
He says because he was working on chickpea, he and this gentleman, Mahendra Thudi, they were working on Mars in chickpea, but they got frustrated. So they abandoned that experiment in chickpea.
Mars.
And they say that when Mars chickpea is not successful, why you are working
No,
on Mars?
I'm not saying why. I'm asking his experience.
Mars, you talk about Mars. What is Mars?
Marker assisted recurrent selection,
Yes, we talk about Mars.
chocolate company.
Talking about Mars. that's a very good question. But when we do a Mars selection, marker assisted selection, depends totally on the marker, how accurate.
Yeah.
So how accurate this marker also depends on how hard work you are. Yeah. That means phenotype. How accurate your phenotypes. If your phenotype is really accurate, the marker is going to be really accurate. That means Mars is going to be really good. So that's why I encourage you very, very early. I encourage everyone here
To go to
spend
the
time,
field,
go to the field.
not in the lab.
Phenotype. That's right. That's right. Because when it depends on the field of people, they tell you it's resistant. Really, it's not.
Vincent is the phenotyper, so he's going to tell now.
Right, it's going to work because Mars is going to become a sneaker. But that was not the question. So if I understand your presentation, so you've got one gene for resistance to aflatoxin contamination. Am I right?
What I'm saying that QTLs, so we identify QTLs, I direct my student and also we talk about it here, try to compare with the diploid sequence, diploid physical map. With that QTL, that region, we did, my student quickly identify one single resistant gene in that region.
Based on the gene
Based on the
annotation.
gene annotation.
I mean, the length of your region is fairly large. No? So
Yes, that's actually I do have that calculation from base pair to how many base pairs.
On the mega basis right now.
Yeah.
Your question? Okay, I got last question, Baozhu. So you have shown fantastic slides, many people working in the field and different thing. When you showed my picture, I was having lunch, but I also visited with you in the field. You did not have that picture. Why you had this picture having lunch? Why the way lunch was great, so doesn't matter. But then you did not take the picture what we were together in the field.
Southern Georgia has a saying. Understand? Southern Georgia. I'm from Southern Georgia. has a saying. Because I'm a Baptist. Baptist likes to eat. Yeah, that's all.
Okay, okay, with these words, well, thank you very much, Baozhu, once again. And thanks to all of you for participating in this discussion. And our colleagues, many colleagues from ESA, they were already watching our jokes as well as that our presentation, your presentation. So thanks a lot to all the colleagues who are participating through online. And with these words, do you have some tea coffee outside? Okay. So we have tea coffee outside, and then some of you, please join us for tea coffee and interact with Baozhu and Steve. And Steve will be giving a talk on Monday. Unfortunately, I will not be there, but many colleagues will be here.
If you want money, talk to Steve.
This is friendship, Baozhu. I'm appreciating that you brought Steve here to Ikri, sir. Thank you very much.